Okay, good morning everyone. Welcome to lecture 26 of BMS 300. Today we're going to continue with part 2 of the muscular system. Alright, so here's an overview of what we were discussing yesterday. We talked about the structure of the skeletal muscle. We looked at the organ level organization. Then we moved down to the structure down at the cellular level. And then we talked about cross-bridge cycle and also excitation-contraction coupling, and that's where we stopped, and we'll pick up from there and continue our discussion. After that, we'll talk about some differences between the cardiac and the smooth muscles in reference to the skeletal muscle. So we won't spend too much time talking about the smooth muscle. I'll just give you a little bit of introduction so you can compare and contrast how it differs with the other two types of muscles, but our main focus will be differentiating between the functions of the cardiac and the skeletal muscle. So that being said, let's move on to excitation-contraction coupling. So I told you that excitation is referring to the signals coming all the way from the central nervous system down to the neuromuscular junction as shown here. And from the neuromuscular junction we have the action potential causing the acetylcholine to bind to specific receptors on the sarcolemma, which is the membrane of the muscle fiber. And the binding of these neurotransmitters will cause the opening of ligand-gated sodium channels, which in turn will cause sodium influx, and there will be this depolarization and the action potential, which is going to travel down the T-tubule. And as the action potential travels down the T-tubule, it causes calcium to be released from the sarcoplasmic reticulum as shown here. And how this happens is that the voltage-gated channels, which are dihydropyridine receptors, they will be activated by the incoming action potential, and they in turn will interact with the ryanodine receptors, which are adjacent to the sarcoplasmic reticulum, and it's through the ryanodine receptors that calcium is eventually released into the muscle cell. And this calcium will bind to the troponin C subunits on the thin filament, leading to tropomyosin moving on the thin filament, exposing myosin binding sites, and then myosin can bind to actin, and that leads to the muscle contraction, and this sarcomere is going to shorten in length. So this is what we discussed yesterday. Now the question is, how does the sarcomere relax? Alright, so in order for the sarcomere to relax and eventually the entire muscle to relax, there are three things which are going to take place. Alright, first, you see that there are no more neurotransmitters here at this neuromuscular junction. And the reason for that is that acetylcholine esterase is going to break down acetylcholine, and so there is no more of the action potential being transmitted down the T-tubule. So that's the first thing which takes place, getting rid of the acetylcholine through acetylcholine esterase. Secondly, calcium needs to be removed from the thin filament, and this is possible through the pumps known as Cerca pumps. Cerca pump is referring to the sarcoplasmic reticulum calcium pumps. Alright, so that's what Cerca pump means. It's the calcium pumps found on the sarcoplasmic reticulum. Now these pumps, they need ATP in order to function, and so they're going to pump calcium right back into the sarcoplasmic reticulum. So removal of calcium means that this thin filament is no longer permitting myosin to bind to actin, right? Because we see that TNI is again going to interact with actin in the absence of calcium, and tropomyosin will eventually move into that blocking state. Another thing also happens, the myosin heads, they will stop binding to actin when ATP binds to it. We see that the myosin heads will remain bound to actin as long as the ATP doesn't bind to it, but the moment ATP binds to the myosin head, it will detach. So these three things lead to the sarcomere relaxing and eventually the muscle relaxing. So let me repeat these three things. First one is the removal of the neurotransmitters. Secondly, you have actin, you have ATP binding to myosin heads, causing it to detach from actin. Thirdly, calcium is going to be taken back into the sarcoplasmic reticulum through the Surca pumps. That's how the muscle relaxes. Are there any questions for me? So pretty straightforward. Yes, no, yes.
So it doesn't take any energy to keep your muscle contracted?
You need ATP to keep your muscle contracted.
You also need ATP to relax.
Exactly. Alright, so I have a real world scenario. When a person dies or an animal dies, after some time the muscles start stiffening, right? So this is known as rigor mortis, right? So the question is, why does post-mortem stiffening of muscles happen, right? So after death, why do the muscles start stiffening? Why does it lead to rigor mortis? You could be able to explain it based on what we just now discussed. All right, so this is a think, pair, share activity. You can turn to your neighbors and discuss, or maybe talk to the TAs, and we'll reconvene back after a minute to see if you can figure out why the muscles start stiffening after death.
Just linked on campus, the muscular resistance slides are the start of the tablet. That's what we're on to. Yeah, that's pretty cool.
I don't like the order, but I think this is the only time I do that I step back.
No, yeah, no. Yeah, it's just different from what I
Okay. I'm not going to call out on any specific TA, but if anyone has ideas, do you want to just share it with the class, or share it with me and I'll share it with the class? Okay. Well, yes. Hey guys, could we have silence? I can't hear. Yeah, go on. Okay, the lack of ATP is preventing sarcomere relaxation. That's one reason. What else? Yes.
Acetylcholine can't be broken down after a certain period of time.
Right. Acetylcholine is not being broken down. Another reason. Anything else? Yeah.
When the muscle cell dies, all the calcium is just kind of released.
Wow. Yes. So calcium is going to flood the thin filament. Excellent. You guys have all hit the 3 points I wanted to talk about. Right. So the three things which cause muscle relaxation are being affected. Right. You don't have the acetylcholine being broken down. That's not the main reason I'm going to talk about, though. We'll see what happens when there's a lack of ATP. So we know that ATP is needed for the myosin head to detach from the actin filament. That's the first thing. Secondly, ATP is also needed for the sarcoplasmic reticulum calcium pumps to function. So for the Surca pumps to work, it needs ATP to move calcium back in. And when the ATP is depleted in the muscle, it causes calcium to flood the thin filament. As a result, the myosin heads which have ATP bound to it, they will attach to actin and undergo the power stroke, and they will remain bound to the thin filament, and because there's no ATP to make it detach. And we even were discussing about what do you need in a relaxation solution in addition to EGTA, which removes calcium. ATP is needed to make the muscle relax. Is this clear, folks? Why rigor mortis happens? So forensically they can determine the time of death by looking at the extent of rigor mortis. So rigor mortis will start at the smaller muscles, like the facial muscles around the mouth, and then the muscles in your fingers and digits. They are smaller muscles. ATP will deplete a lot quicker, and it's later on that the larger muscles will start tightening as well. So around 6 hours after death, rigor mortis will start, and by 24 hours it would be done. And after 36 hours, the body will no longer be stiff. Any guesses on why rigor mortis will eventually end? Yes?
All of those things, keeping things flexed or are just being used, so it'll all be depleted eventually.
Well, the reason rigor mortis will eventually end and the body becomes flaccid is because the proteins start degrading after a few days. Right, after a couple of days, the proteins will start degrading, so they're no longer muscles are no longer contracted. Right. Is that clear, folks? Okay, great. So now we are going to shift gears from the skeletal system and move on to the skeletal muscles and move on to the smooth muscles. All right. Yeah. So smooth muscles. One of the main things to consider is that it is under involuntary control. Right, so we see that smooth muscles will be in your blood vessels, as shown right here. It can also be lining the various body cavities, like your digestive cavity, around the stomach, around the esophagus, and there's no way we can really control the smooth muscles. And we also find that the smooth muscles will be found in the eyes as well. That will help the constriction and dilation of the pupil. So yesterday when we were talking about the autonomic nervous system and the activation of the parasympathetic division, that's what is necessary for the pupils to constrict. Let's say you're trying to focus on something, maybe you're trying to read some really small lettering, the pupils will constrict due to the smooth muscles in the eyes. On the other hand, let's say you have sympathetic activation, you see something scary, it's very hard to focus on small details because your pupils start dilating to allow more light to come in, and such. In a time like that, it's very hard to focus on minute details. Okay, so these are just examples of places where you have smooth muscles, and we find that the shape of these smooth muscles are spindle shaped. Right? It's not like the skeletal muscle, which is really thin and long. It's spindle shaped, and the length will be around 300 to 200 micrometers. So it is much smaller in length than the skeletal muscle, which can extend the entire length of the muscle, even up to 30 centimeters. We also notice that there's only a single central nucleus in smooth muscles, in contrast to skeletal muscles, which have lots of, it's multinucleated. There are no tendons, no T-tubules, myofibrils, or sarcomeres. So we know that it is the sarcomeres which are necessary for the striated pattern in the skeletal and cardiac muscle, but here you have no sarcomeres. And now the question is, how do the muscles contract if they don't have any sarcomeres? And so even in the smooth muscles you have thick and thin filaments, and if you notice this figure here, the thick filaments are shown in green. So those green lines in between, they are the thick filaments, and the purple lines in between those green lines, they are the thin filaments. So we see that the thick filaments are scattered, and you also have these thin filaments overlapping it. And in the skeletal muscle, the thin filaments were linked to the Z line. Here there is no sarcomere, so there's no Z line. Instead, these thin filaments are linked to what is known as dense bodies. So those circles you see on either side, they are the dense bodies. And each dense body is linked one to another by intermediate filaments. Right? So we see that among the cytoskeletal proteins we discussed, intermediate filaments will offer more mechanical strength, and it's the intermediate filaments which link the dense bodies. And so when calcium comes into the smooth muscles and eventually it leads to contraction, we see the thin filaments will slide over the thick filaments, and the single smooth muscle cell can decrease by 70% of its original size. So when it's relaxed, it will be at a certain length. When it contracts, it can decrease by 70% of the original length. So this panel on the left is just showing you what's happening at a level of a single cell in the smooth muscles. But if you were to look at the panel on the right, we see that these dense bodies, they are connected even between the two cells, right? And so when one cell is contracting, it's going to pull on the neighboring cell as well, and together the entire tissue will contract when it receives the stimuli. So this is why there are no striated patterns in smooth muscles, because you don't have ordered myofibrils. You have there are no Z lines, instead there are only dense bodies to which the thin filaments are linked to one another. Does this make sense? Yes? Okay. So how is smooth muscle contraction controlled? Now in smooth muscles you don't have the neuromuscular junction, right? Instead we find that there are a number of neurons which will innervate the smooth muscles, and from these neurons the axons which are in proximity to these smooth muscles, they have these bumps known as varicosities. So there are these little swellings in the axons called varicosities, and inside these varicosities you have the neurotransmitters such as acetylcholine. And so when a signal from the autonomic nervous system reaches these neurons, it's going to cause the neurotransmitters to be released from these varicosities, and acetylcholine is going to bind to ligand gated channels, which will eventually cause a change in the voltage, right, and that will cause the opening of voltage gated calcium channels, and calcium can enter into these smooth muscle cells and it can lead to muscle contraction. Okay, so there are no neuromuscular junctions, there are just these varicosities containing the vesicles which will release the varicosities containing the neurotransmitters, which when released can lead to calcium entering into the smooth muscle cells. All right, so let's consider how calcium is going to bring about a contraction of the smooth muscle cells. So what we're going to consider now is just a very high level view, and contraction in the smooth muscle is a lot more complicated, but I just want to give you a few steps, right, for the scope of this course. Now what you see here is the actin monomers which make up the thin filament, right, and here is a myosin head which will be found in the thick filament. What is the one thing which is missing in the thin filament? Yes? The ATP is, there's no ATP shown here. What else is not shown here on the thin filament? Yes? Tropomyosin and the troponin complex. So you don't have troponin and tropomyosin in the smooth muscles, right? So the question is, how is calcium going to bring about muscle contraction here, and also what is regulating the interaction between the myosin head and actin? That's the question. Now if you notice the myosin head, it's got this little box next to it with the letters RLC, and RLC stands for regulatory light chain, right? So this light chain is going to have some regulatory function. Why is it called light chain? Because it's much smaller in size compared to the myosin molecule, right? That's why it's called regulatory light chain. So calcium is going to enter into the smooth muscle cell, and it will bind to a protein known as calmodulin. Okay, and what calmodulin does is calmodulin is going to activate the myosin light chain kinase. So myosin light chain kinase is an enzyme which is going to add a phosphate group. That's what kinase means. It's an enzyme which will phosphorylate certain amino acids, and when this kinase is activated, it's going to add a phosphate onto the regulatory light chain. It will phosphorylate the regulatory light chain, and when the regulatory light chain is phosphorylated, it's going to interact with actin, and it can strongly bind and produce the force. And notice here that I haven't shown ATP in this slide, but ATP is used by these myosin heads for the contraction of the smooth muscles as well. Just for the sake of simplicity, I haven't shown ATP in this cartoon. Now because phosphorylation was what brought about muscle contraction, dephosphorylation will bring about smooth muscle relaxation. And there's another enzyme for this. It's called myosin light chain phosphatase, which will remove the phosphate group, and that will permit the myosin from interacting with actin, and that leads to relaxation of the smooth muscle. Are there any questions at all on how contraction takes place in the smooth muscles? So whenever you have flowcharts like this, it's very easy to write a critical path question, right? If you were to affect one part in this process, how will it affect muscle contraction, right? So please make sure you understand these flowcharts well. Okay, so now let's talk about how control of contraction in the smooth muscle takes place. We see that there could be two types of units. The first one on top here is the multi-unit smooth muscle, and both are just the same picture. I'm just using two different pictures to highlight different features, right? So the top one is the multi-unit smooth muscle, and the one below is the single unit smooth muscle. So in the single unit smooth muscle, you have a single motor neuron innervating a number of smooth muscle cells, right? So in a single unit smooth muscle. Whereas in a multi-unit smooth muscle, notice that each one of these cells, they are innervated by different motor neurons. Right? So in multi-unit smooth muscle cells, each one is innervated by a motor neuron. And where you find this multi-unit smooth muscle cells will be in parts of the body such as blood vessels, eyes, the bronchi, and these smooth muscles, they are going to be under hormonal control. Right. So, for example, let's say you have the sympathetic division being activated, and you have epinephrine being released. That can cause these smooth muscles in the blood vessels to either contract or dilate as needed. And when you think about these single unit smooth muscle cells, we notice that each of these cells, they're going to be connected one to another by gap junctions. Right, so these cells are connected by gap junctions, and so even though only one cell is receiving the acetylcholine, right, and that causes calcium to come in, we see that calcium ions can flow from one cell to the other. And the unique feature about these smooth muscle cells is that they are activated by stretch. Right, and we can think about different parts of the body where stretching the muscle will lead to its activation. So one example would be the digestive system, and let's say the stomach is also surrounded by smooth muscles, and as you're eating, the stomach contents are going to increase, and it will cause the walls of the stomach to stretch. That can activate these muscles, right? And we see that as the stomach stretches, signals can be sent to the brain to say that you've eaten enough, stop eating. And we will take a closer look at this in the digestive system. We also find that in the urinary bladder too, signals can be sent to the brain as the bladder stretches, as it's being filled with urine, so that the brain knows that it's time to urinate as well. So that is all I have to say about the smooth muscles. I don't have anything else to add, but we see that there are some features which are similar between the different types of muscles, and smooth muscles are uniquely different because they don't have the sarcomeres. Yes?
How is it that we're able to control our breathing?
So breathing, there are skeletal muscles like your diaphragm and so on, so that's how you have certain parts which can be controlled. Yeah. And we will get into more details of that when we discuss the respiratory system, on how breathing control happens. Any other questions? Okay, moving on to the cardiac muscle. I want to first point out the main structural similarities, and that is the presence of ordered myofibrils. Right? So we see that you have sarcomeres in the cardiac muscle as well, and this is the reason why it has the striated appearance, and these myofibrils are going to be well ordered. Now let's look at some of the differences. We first see that these cells are going to be much smaller. The length is going to be 50 to 100 micrometers, and the width around 10 to 20 micrometers, whereas the skeletal muscle cell, the diameter alone is going to be around 100 micrometers, and the length would be for the entire muscle. And these small cardiomyocytes, they're also going to have a branched appearance, and this branching appearance along with the small size will permit the muscles of the heart to contract in unison, right? And so we see that the contraction and relaxation is going to happen as a single unit, and that allows blood to fill and blood to be pumped out as well. And that's why this smaller length of the muscle cells and also having a branched pattern permits that. In contrast, when you think about skeletal muscles, it's pulling in one direction, right? And so that's why it doesn't have the branched appearance, but it has a linear and a straight arrangement. Notice also that the T-tubules are shown here in the cardiac myocytes, and they're going to be a lot smaller, and so will the sarcoplasmic reticulum surrounding the surrounding the myofibrils. They're going to be a lot smaller as well. The exact relevance of this, it's beyond the scope of our discussion, but just know that the way cardiac muscles are sensitive to calcium compared to skeletal muscles is going to be slightly different, right? And I'm happy to discuss those details with you after class, right? But for now, just know that these structures are much smaller. We also notice that there's a lot of mitochondria in the cardiac myocyte, and we will talk about how the body is making ATP from different macronutrients later in today's lecture and also in Monday's lecture, and we'll understand why the cardiac muscle needs to have more mitochondria, because it has a more of endurance activity it is performing compared to skeletal muscles. We also find that these cardiac myocytes, they're going to be linked one to another by these structures known as intercalated discs. So what you see right here labeled in red, it's known as an intercalated disc, and the intercalated discs have two types of cell junctions, right? It has desmosomes, and we talked about desmosomes in which other organ system previously? You guys remember? Integumentary system. And why was it important to have desmosomes there? Say that? Cell to cell connection, but why are desmosomes so relevant for the integumentary system? Yes.
How they respond to mechanical stress.
Yes, desmosomes can withstand mechanical stress better because they are linked to the intermediate filaments, right? And so we see even in the cardiac muscle, it's going to undergo a lot of mechanical stress because it's constantly pumping, and the cardiac muscle never takes a break, starting from 21 weeks until the end of a person's life. So desmosomes offer the mechanical support. Another kind of cell junction is found, and these are gap junctions. And gap junctions, they couple cells electrically and metabolically, right? That's something we talked about in Unit 1. And as far as the cardiac muscle cells are concerned, calcium ions can flow from cell to cell, and it can bring about muscle contraction. So ions flowing from cell to cell leads to muscle contraction. So I will get into more details about this in the cardiovascular system, but for now I just want to talk about the conduction system of the heart. So we see that the heart has got two types of cells. You have the cells which are capable of contracting and relaxing. Those are the contractile cells. And you have a second type of cells which is only going to make up 1% of the total number of cells. Those are known as autorhythmic cells. These are cells which are capable of generating their own action potential. And where you find these cells are along the conduction system of the heart. So you'll see them in the sinoatrial node shown here. You'll find them in the atrioventricular node. And notice that these two nodes are connected by an internodal pathway. So there also you'll have these cells. And then you'll find that there will be these two branches which come off from this atrioventricular node, and it gives rise to these fibers known as Purkinje fibers. So all of here you have the autorhythmic cells. And so these cells will generate an action potential which is going to rapidly have the current passing through these cells, and from these cells it will enter into the contractile cells via the gap junctions. So in the conduction system, and because of the gap junctions, the action potential is going to rapidly propagate throughout the entire myocardium, which is the muscular part of the heart, and it causes the cardiac muscles to contract and relax as a single unit, right? So it's not only those small branched cardiac myocytes which permit this, but also the fact that they are linked through gap junctions. So this permits the heart muscle to contract and relax as a single unit. So with that, I'd like to summarize what we were discussing from yesterday onwards. We talked about the different layers of connective tissue surrounding the various parts of the muscle. So we see that the muscle is surrounded by the epimysium, and then the bundles or fascicles, they're surrounded by perimysium, and then each myofiber is surrounded by the endomysium. And all of these layers of connective tissue will eventually join to form the tendon, which will link to the bones. And we looked at some key differences between the skeletal muscle and the typical skeletal muscle cell and a typical muscle cell, and this includes the length, the number of nuclei, the types of organelles inside it, like sarcoplasmic reticulum and the ordered myofibrils. Those were some key differences. And then we saw how contraction and relaxation takes place at the molecular level and how it translates all the way to the organ level. And we talked about that by looking at what happens in the sarcomere. You have thick and thin filaments in the sarcomere. They will interact with one another, and that leads to contraction. We specifically saw that in the thin filament, it can exist in 3 states depending on the troponin-tropomyosin complex, how its structure is in the presence and in the absence of calcium. And we also find that these 3 states of the thin filament, they're going to align with the crossbridge cycle, where the myosin heads can exist in 3 states: the unbound state, weakly bound state, or the strongly bound state with actin, which produces force. And then we talked about excitation-contraction coupling, which we revisited today, and then we saw how relaxation takes place as well. After that, we spent a little bit of time talking about some of the structural differences between smooth muscles and cardiac muscles, and compared it with the skeletal muscles as well. All right. Are there any questions for me? Yes.
So, for cardiac muscles, and more specifically cardiac arrhythmia, is that caused because the action potentials aren't propagated like at the same time between the blood and left atrium?
So the question, what causes cardiac arrhythmia? We'll talk about it in the cardiovascular system. Short answer is an electrical problem. So the passage of the action potential is affected, and depending on which part of the conduction system it's affected, you have different classifications as well. We'll briefly touch about it in the upcoming lectures. You seem very excited. Yes.
So, what is there like an actual purpose of the closed state on the
crossbridge cycle? So what is the purpose of the closed state? So to transition from the block state to the closed state, you need calcium to do it, but the closed state is not sufficient to produce force. It has to transition into the open state, which is brought about by cross bridges. Right? So it's when you have more steps, it's offering better sense of regulation of how force is produced. That would be the short answer. Happy to discuss more about it after class, because there are some conditions where you have mutations to the proteins which bring about calcium binding, which can affect the closed state as well. But for the scope of our discussion, know that it's just one of the intermediate steps before it goes into the open state. And even this 3-state model is a big generalization. There are models which talk about 16 states and so on, but for the sake of our discussion, we'll just keep it to 3 states. So it makes sense? Yeah. Okay. Anything else, folks? Okay, so if you have no more questions for me, let's quickly look at a couple of polar viewer questions. Which of the following connective tissue are tendons composed of? We talked about this yesterday. Some of you gave me the right answer. Let's see if you still remember this. Right? Areolar connective tissue, dense regular connective tissue, reticular connective tissue, elastic connective tissue. Okay, we have 61 responses. It's okay. I won't bother the TS for now. Okay, most of you said dense regular connective tissue, and that is correct. Okay, it is dense regular connective tissue which make up tendons. Okay, here's another question. You may want to talk to your TS for this. Right? So the intracellular concentration of blank is higher than the extracellular concentration. So this would be the case in both muscles and in nerves as well. So I hope you're able to answer this question. Is it sodium ions, calcium ions, potassium ions? Which one would have a higher intracellular concentration? If you want a hint, think about the action potential and what happens during the various phases of the action potential, and that should give you an idea. Okay, Ronan, do you want to pick someone from your group to give us the answer? Was that okay? Potassium. Most of you said potassium. For the one person who doesn't know, the right answer is potassium, right? So I don't want you to memorize these numbers, right, as far as the concentration of these ions, but just know that calcium and sodium will be higher in the extracellular fluid, right, and potassium ion concentration will be higher in the intracellular fluid. So in the action potential, right, during the repolarization, what is leaving the cell? It's potassium ions, because there's higher concentration of potassium inside the cell. And also during the depolarization phase, you have sodium ions flowing in because it's got a higher concentration in the extracellular fluid. And calcium entering into the cell is very tightly regulated, and that's what leads to muscle contraction, right? So if you have high levels of calcium inside the cell, the muscle regulation really cannot happen. Okay, is this clear, folks? The functional relevance of why these three ion concentrations are different. I don't need you to remember these numbers, but I hope you know in terms of which one is higher on the outside versus the inside. Sounds good? Right. Okay, any questions? If there are no questions, we are going to switch gears and talk about energy, metabolism, and muscle performance, right? So how many of you have heard the term Krebs cycle? Okay, most of you have. Okay, great. So for many of you this would be a review, right? But let's lay a few foundational concepts down, and then we'll talk about how it can influence muscle performance. So here's an overview of what we'll be discussing, right? So we are going to answer the question, how does the body extract energy from food? So do you remember what are the three macronutrients? Anybody? Fats, lipids, right? Yes, carbs, lipids, and proteins, right? So sugar, fats, and proteins. Those are the three types of macronutrients. So these aerobic and anaerobic pathways are going to be used to extract energy from carbohydrates, right? So that's the first pathways. We'll be looking at the aerobic and the anaerobic pathways. Whereas when it comes to fats, beta oxidation is a very important step for making ATP from fats, and deamination is something which will be used to break down proteins. So the amino acids will have an amino group that will be taken away by deamination. So this is the first question we'll be answering. And then later on we'll be talking about the connection between metabolism and respiration. So when you think about metabolism, this is something connected to the digestive system, where we get nutrients into our body, and then from there ATP is made. It can also be linked to skeletal muscle function, where nutrients can be broken down to use the ATP as well, to make and to use ATP. And respiration is of course linked to the respiratory system, but what we see here is during metabolism that all of these systems are going to work in a connected manner. So we lay down the foundations at the cellular level. What is the link between the digestive and the muscular system, and also the respiratory system as well. And then finally we'll talk about what fuels the body will use depending on the type of activities. So some activities are endurance activities, others are high intensity, short duration activities, and depending on the activity, the kind of fuel used can be different. And we'll also see how there are different types of muscle fibers which use different types of fuels to make ATP. So with that overview, let's talk about what do we mean by metabolism, right? So metabolism is referring to all of the chemical reactions which are taking place in an organism to maintain life. So these are a whole set of chemical reactions. Now these chemical reactions can be broadly divided into two types. You have catabolism and anabolism, right? So catabolism is where these reactions are going to break down molecules and use the energy in those molecules to make ATP. So that is catabolism. And then anabolism is where these reactions will synthesize new building blocks, right, to produce new molecules. So they're going to synthesize building blocks to make new molecules. So to give you examples of both these types of reactions, I want to draw your attention to this cartoon here, right? So on the upper panel, you see glycogen being depicted, and what glycogen is that it's a polymer of glucose subunits. So all of these glucose subunits are linked to one another, and that is glycogen. So let's say you go out and exercise, right, and we see glycogen could be in the liver, it's in the muscles, and as you exercise, this glycogen is going to be broken down into these glucose subunits, and those glucose subunits can be used to make ATP. So this breaking down of glycogen into glucose is an example of catabolism, right? And then after the exercise, you go and you eat a carb-rich meal. The glucose which you ingest is going to be stored as glycogen in the muscles. So usually the amount of glycogen stored in your muscles will be slightly more than what you had before the exercise, right? So that's how the body stores the glucose. But let's say you eat way more glucose than your body needs, right? So what happens is that all of the excess glucose is going to be stored as what? Fat. It's going to be stored as fat, right? So that's also another example of anabolic reactions. So our goal is mainly going to be looking at catabolism in this series of lectures to see how the muscles are going to use these nutrients to make ATP and eventually carry out muscle contraction. So before we proceed, I want to point out how is energy found in these foods which we get from plants, and so I want to highlight the process of photosynthesis. So we see that there's carbon dioxide in the air, and so there's water as well in plants. They can use the energy from the sun to make carbohydrates, right? So photosynthesis is the process by which the green plants are using energy from the sun to make carbohydrates using carbon dioxide and water, and oxygen is also going to be released as well, right? And we see that energy from the sun is required for this because bringing these carbon-carbon bonds together requires a lot of energy. So in these carbon-carbon bonds there is chemical energy. And when we eat plants or when we eat animals, we are using the energy in the carbon-carbon bonds to make ATP, right? So our bodies is going to extract the energy in those carbon-carbon bonds to make ATP, and ATP is the energy currency which our body can use. So how this energy is used to make ATP is that this phosphate group is going to be added on to this ADP molecule. Where there are two phosphates, this third phosphate is going to be added on, and doing that requires energy because we see that here's a cartoon of the adenosine triphosphate. To add the first phosphate, you need a little bit of energy. Adding the second phosphate is going to require a lot more energy because both phosphates are negatively charged, and like charges will repel each other. But adding this third phosphate is going to take way more energy because these two phosphates are repelling the third phosphate as well, because like charges repel. And the analogy of how our body uses the energy in these phosphates to carry out various activities, similar to when you draw back a bowstring, right? And so the energy you put into drawing back the bowstring is going to be used to shooting the arrow forward. Similarly, the energy it was put into making this ATP is going to be used to fuel various activities. So let's talk about how energy is extracted from carbohydrates. So there are four pathways or four metabolic pathways we're going to consider to see how energy is absorbed from carbohydrates. The first one is glycolysis, where glucose is going to be broken down into pyruvate. So that's the very first step. Second step is pyruvate will be converted into acetyl-CoA, and thirdly, acetyl-CoA is going to enter into the citric acid cycle, which takes place inside the mitochondria, and we see that a lot of high energy electrons are going to be released in these above mentioned steps, in glycolysis, in the conversion of pyruvate to acetyl-CoA, and in the citric acid cycle. You have high energy electrons being released, which will be taken to the electron transport chain, and that energy can be used to make ATP. Right, so this is the high level view of what's going on in extracting energy from carbohydrates. So what I would hold you responsible for is that just know what is the substrate entering a specific pathway and what is the product. Right, that's the first thing. Secondly, I want you to keep track of the number of carbon atoms. Right, so first one is know what's the substrate entering in, what's the product, keep track of the carbon atoms. So let's talk about glycolysis. So the term glycolysis is referring to glucose splitting, because we see here you have 6 carbon atoms in glucose, and if you break this into half, you will have 2 molecules each having 3 carbon atoms. So you start with glucose with 6 carbon atoms, and you end up with pyruvate, which has got 3 carbon atoms each with 2 pyruvate molecules. And we see that this does not require any oxygen. This series of steps, it's actually going to have around 10 different steps happening here, and it does not require oxygen, and it's going to take place in the cytosol. Notice here that you have 2 ATP molecules being invested early on in the reactions, and later on you only get 4 ATP. So if you think about it, there's only a net yield of 2 ATP. You get very little ATP, but the rate at which this ATP is produced is quite fast. Right? Notice also there are these high energy electrons which are released in these series of reactions, and these high energy electrons will be carried by an electron carrier, NAD+. Right? And what is the full form of NAD+? Not required for our level of discussion. Right? There's also another electron carrier we'll talk about later on, but we see that these high energy electrons will be eventually taken to the electron transport chain. Now pyruvate, which is shown here, can have 2 fates. Right? It can either be converted into acetyl-CoA, as I mentioned previously, or it can be converted into lactate. And what determines whether it turns into lactate or acetyl-CoA is the amount of oxygen. Right? So if, let's say, you don't have as much oxygen available, then it's going to convert it into lactate, and lactate can be moved to the liver. Right? So here's a cartoon showing you glucose being converted into pyruvate, and that's turned into lactate, and lactate goes to the liver, where it is going to be converted back into glucose, and the glucose is going to be sent back into the bloodstream to go to the muscles. And the key here is, notice that there's so much of ATP being used in order to convert lactate back into glucose. And you may be wondering, why is this happening? Why are we not being energy efficient, right? And let me end by giving you this analogy. So let's say you're trying to get from here to DIA, and you tell your Uber driver that I'm running late, I'm going to double what I'm going to pay you, right? And then you get to I-125 and you see there's traffic backed up, but you say I'll pay you way more, right? I'm going to triple it, because you don't care about the price being paid, you just want to get there soon. And so when you have a high intensity activity and you're trying to escape from somewhere, the body would rather use this mechanism to make sure that ATP is generated quickly rather than slowly. Alright, so I'll let you go now, and we'll talk about conversion of pyruvate to acetyl-CoA when we come back on Monday. Thank you all for your time. Have a great weekend. And I'll be releasing the review slides for exam 2 either today or tomorrow, but it will be released tomorrow at the latest.
Thank you. That's what I thought. Like, he's like, oh no, do I have to